Welcome, everyone. My name is Renee Duprel, and I'm the Director of Development here at the Institute for Systems Biology. Thank you all for joining us for our almost monthly Research Roundtable. This is our virtual series that we host to keep you up to date on research at ISB. This series is geared towards non-scientists, and our goal is to make our work more approachable to all. Our intention is to have one of these just about every month, so keep an eye out for our emails. Today, we'll be hearing from our featured scientist and his guest. Then there will be an opportunity for questions and answers after that. Today's topic, DNA sequencing in the high school classroom. There's a Q&A box at the bottom of your screen, and you are welcome to submit questions at any time. I'll be monitoring those, and at the end of the presentations, we'll move to the question and answer session, and we'll work through those questions. Chris Lausted is a senior research engineer here at ISB. He works on multi-omic instrumentation and data analysis. He's been with ISB since our founding in 2000, and his research interests include single-cell multi-omic analysis of complex diseases like metastatic cancer and macrocytic anemia. More recently, he has been working on collecting and integrating deep phenotyping data sets to characterize the transitions that occur with acute and chronic COVID-19. Chris is joined today by Dr. Danielle Vermaak. Danielle grew up in South Africa, then moved to the US to get her PhD in molecular biology from Johns Hopkins University. She made a career change to education, and she's now teaching chemistry, physics, and a new course named Genome Science at Lincoln High School, a public school here in Seattle. Welcome, Chris and Danielle.
Thanks, Renee. I'll share my screen now. Okay, I hope everyone can see that. So yes, I'm Christopher Lausted, and I'll be co-presenting today with Danielle Vermaak of Lincoln High School. So we're a husband and wife team leading an effort to bring modern DNA sequencing to the biology classroom. We're going to tell you about a curriculum pilot project that we developed recently and then tested over the course of about a month at Lincoln High School in their new Genome Science elective class. I think it was quite successful, and so we're going to be working to share this curriculum and to try to put together kits to make it easier for other schools who might wish to use it. So from its very inception, education's been a top priority for ISB, right alongside research. For example, our Logan Center provides services like professional development for teachers, and our Systems Education Experience Group develops classroom curriculum. In addition, pretty much all of our scientists and staff here participate with education groups, working with them, serving as mentors to our many high school and college interns. As an engineer here at ISB, I've been working on providing things like hardware and software support for some of these new curricula and materials for laboratory experiments. So a few years ago, Becky Hausman, one of the members of the SEE, and Danielle and I were brainstorming and trying to think of a way to perhaps bring a real-world hands-on genomics experience to the classroom. And if we could make it work, Danielle had a new genome science class where we could test that. Now, the reason we thought that this was important is that today, DNA sequencing is everywhere. It's fundamental to biology. The fundamental unit of the biology is the cell, and a cell is largely defined by its genome. But DNA sequencing has all sorts of real-world applications. It's not just used in research laboratories like our own or Swedish and Providence hospitals. We're part of that network. It's also used by public health, especially now in the time of COVID, for identifying and tracking variants. People are very interested in genomics now with companies like 23andMe, Ancestry, Genealogy, etc., to look at family histories and genetic disease predispositions. So this is something that really affects all of us. It's really not an esoteric topic anymore, but high school AP biology and biotechnology labs really haven't incorporated yet. They mostly remain with more traditional low throughput DNA, RNA, protein related laboratory activities. When sequencing is a part of a classroom, usually that DNA is sent out to a third party. So the actual hands-on experience with DNA sequencing hasn't really appeared yet. And this is because of previous barriers to sequencing in the high school. Teachers, mentors, lab technicians need to be familiar with the topics. They need to be familiar with the methods. And they need to be able to get equipment and consumables, which up until very recently have been extremely expensive. But this changed quite recently. So when ISB started 20 years ago, we had a huge sequencing group working on chromosome 15 for the Human Genome Project. And we had a lot of these machines here on the left, the first generation of sequencing machines, like the ABI 3000 series. We had tables and tables of these. They were massive, they were heavy, they were extremely expensive. But this is what did the original Human Genome Project. This technology was actually developed by our founder, Leroy Hood. The output at that time, with a team of people running this 24 hours a day, is maybe 0.01 billion bases a day. And these machines were expensive. I won't tell you how much they cost to buy, but just the service contract on it, the annual service contract was about $50,000 a year. Now, sequencing's gotten a lot cheaper, mostly because the throughput has gone way up. So today, our institute uses the second generation of sequencers sold by Illumina Corporation and a few of their competitors. So for example, the next Seq machine that we have, it's still pretty massive. It's still 100 kilograms. The service contract is still almost $50,000 a year, but the output has gone way up to 100 billion bases a day. So it's really impressive how much data it can produce, which makes the cost per base a lot less than it used to be, which is why you hear about how a human genome can be done for maybe $1,500 or even $1,000 if you get all the discounts. Still, this is something that takes a lot of space and money to get started. It's not something you can do at home. Well, there's a new company with a third generation sequencer, Oxford Nanopore, that has a device that it looks a little bit like a stapler here. It's only 400 grams. It's about the size of a small stapler or a really large USB stick. And it doesn't have any service contract. It's basically like an inkjet printer. If you buy the ink cartridges, they give you the printer / sequencer for free. So there's no service contract. There's really no capital investment. You're just paying for the consumables. And its output is a pretty impressive 10 billion bases per day. So within striking distance of the big expensive machines. And the data quality is pretty good too. So it works by a fundamentally different technology. Instead of using fluorescence attached to the bases A, C, G, T, and doing degradation of the DNA or synthesizing the DNA is run unmodified. The DNA runs through a nanopore. So this is a pore that was taken from E. coli and stuck into a membrane, and then electrodes were put on each side of that membrane. So the only way that anything can get through the membrane is through this protein pore. Usually ions go through it. Salt ions go through when the electrode is turned on, and you can measure how fast the ions are traveling through the pore. Now, when something gets stuck in it, like a DNA sequence, a DNA strand, it fills up part of the pore and it slows the current flow through the pore. Now, the 4 bases of DNA, A, C, G, T, are all slightly different sizes. So there's a unique signature, a unique current as each base passes through the pore. And these things pass through at hundreds per second. So in a few seconds, you get thousands of bases of DNA. It also can read extremely long stretches of DNA. So the previous technologies could read out maybe 100, 200, at most 300 bases of DNA. These have no trouble with 5,000, 10,000 bases. We've gotten 100,000 bases. Some people have even seen 1 million bases of DNA in one strand go through one of these pores. So it's really a unique technology. Now, it's cheaper for a flow cell. It's still pretty expensive. So we started using this about 6 years ago, and we would pay $900 for a flow cell. So to run a sample was $900. That's still a little bit expensive for home use or for classroom use. But then 2 years ago, they decided to come up with a smaller flow cell. So instead of having 500 pores on the flow cell, they just put 128 on. But what was really nice is they reduced the price then to just $100. So now imagine a group of 4 people get together and they can pool some samples, and for $25 each, they can start a sequencing run. So now we're actually getting into sort of the DIY home or classroom scale. Well, we wrote a few grants. Danielle was the most successful. Toshiba was interested in supporting high school science teachers, and they were really looking for experiments in the classroom, enhancing the classroom laboratory experiments experience, and they wanted something that was really teacher-led. And so Danielle wrote a plan in order to make this a project-based learning experience for the students, where the students would come up with projects that could be answered with DNA sequencing, and they would be able to then get hands-on experience and be able to from firsthand experience, explain how high throughput technologies, DNA sequencing technologies, can answer previously unsolvable questions. So the plan is to teach the technology and then have the students prepare posters where they would explain how they came up with a question, carried out an experiment, analyzed the data, and then they would share this with their peers and then share this with the public. So Toshiba was pretty excited about this and funded it. It wasn't a large grant. The very maximum they could give was $10,000. Fortunately, with the new cheaper flow cells and a few other reagent substitutions we came up with a way to do this for 2 classrooms, 64 students in groups of 4 to each do their own sequencing project. So ultimately half the cost will be the sequencing flow cell, and half will be DNA extraction, getting the DNA out of the sample. So we work to bring down both of those costs as much as we could. There is a one-time cost for hardware to get started. So we need these little adapters so that we can use these cheaper flongle flow cells. And then we also need a computer to do the base calling. It's actually computationally intensive to do this. And so we didn't want to spend $5,000 on Oxford Nanopore's proprietary computer, but instead we budgeted for some $400 NVIDIA GPU-enabled prototyping boards, and basically made the same thing. So with this one-time cost, we could get started, and we could test out a few different alternatives, and then test the curriculum in the class. Now, the genome science class had a few things already available for us to use. They're already doing DNA amplification via PCR. They're running gels. They've got pipetters. They've got mini centrifuges. We also were able to borrow materials from the Fred Hutchinson, such as this fluorimeter here. And then we also managed to find a few home and hardware store type substitutes for some commercial equipment that we would normally need. So here's the other equipment that we had on hand, and then we got started. Well, we ran some tests ourselves, and then we introduced it to the students. So for that, I'll turn things over to Danielle.
Hi, everybody. I'm really excited to be here to tell you about this project. So as Chris mentioned, we really, and Toshiba actually was looking for this as well. We really wanted to work on the curriculum aspect of this grant so that students could learn not just about cool and amazing new technology, but within the context of authentic project-based learning. And throughout the whole project, there wasn't a day where kids weren't saying to me, we're really doing science, right? This is what scientists do, because they can tell the difference. And that is what I believe really made this project such a success, this first run through. So at Lincoln High School, we're actually at a large public high school, but our vision is to engage students in curriculum that involves problem solving and design, and producing work that goes beyond producing grades. And so grades is usually the ultimate goal, but that's not very motivating, and school starts feeling disconnected from life. And so especially in Seattle, with all the amazing opportunities to collaborate with scientists, for example, at ISB, we want students to be experiencing the process of doing science in the classroom and walking away with skills like critical thinking and collaboration instead of just things that are in textbooks. So we really believe that can be done through project-based learning, and then also sharing our learning publicly with an authentic audience. So we broke the project up into 3 parts, and we actually had the luxury of having flexible timing because this project did take quite a large segment of the semester in the end. And the first part was for students to be introduced to DNA sequencing technology and develop their project proposals. So what they produced there was a 1-page grant proposal. And it had to be hypothesis driven. It had to have some kind of community value or interest to them. And it had to be authentic. And by that, I mean, students researched what had already been done, what was known. Things they, for example, came up with was, well, lots of us swim in Green Lake. You know, we heard there were bacteria in Green Lake. What's going on with that? So, and of course, being students, of course, they also all wondered, well, what about my cat and my dog in the same house? Do they have different microbiomes? So we really started with these. This is not a project that was done with students who were very advanced in biology or anything. They've all taken chemistry, physics, and biology, which is a requirement of Seattle Public Schools. But only a few of them have taken AP biology. This project can be applied across a large variety of income of levels of students that come into the class with different backgrounds. And we did take a deep dive into really understanding the methodology. So it is not just a kind of cool toy. So students use things like paper models to learn about chain termination sequencing, Sanger sequencing. And then, of course, having the wonderful connection to ISB and Lee Hood's lab, who, of course, worked on the human genome in the beginning, and of course, the invention of sequencing machines and such. We went through that as well and learned about that. And then we really did a deep dive into whiteboarding and comparing and contrasting how these different sequencing methods work, because it's one thing to mention sequencing gigabases at a time, but to wrap your head around that is quite hard. And so scale and proportion is something we kept coming back to. In terms of microbiome also, there's such interesting things that you can bring into the classroom. We read several excerpts of Ed Young's book, I Contain Multitudes, about microbiome. We used online seminar or Q&A sessions from video series like this one, where we had Sean Gibbons, who works on the microbiome at ISB, of course. We watched some of his videos, and so students were just very excited and deeply understood what was going on with these methods. So there they are explaining sequencing to each other. And then in the second part of the project was then more or less the lab part. So what students produced there were individual lab notebooks. So we made them some nice lab notebooks, and they learned how to actually keep a lab notebook, what to write down, what to document, writing all your thinking and troubleshooting and data. And then we also, so students had learned already prior to the project how to use micropipettes. And I think that is pretty essential because some of these reagents are quite expensive and well, they for a school, they're expensive. And so students having some practice with PCR and being comfortable with micropipetting, I say would be really important. And we still had quizzes and things to assess them throughout. In the last part then, so let's see. So they did DNA isolation, they did PCR amplification to check their libraries, they ran gels, and they also then had to multiplex some samples to run them on the nanopores. And we finally then, it was the exciting day, which Chris can tell you a bit more about, or days where we ran our flow cells. And the third part of the project was actually something that was very surprising to students, that here we have all our data and look, we're not done by any means. So such a large part of science is analyzing your data and evaluating your data and finding ways to communicate it and thinking about what's up next. And so that was really the third part of our project then, where we produced professional looking posters. And we had Claudia Ludwig come in as well as Chris, and to come and do a feedback for students. And then they then presented it just recently at an exhibition of learning. Next slide, please. So all in all, students collected 48 samples, basically in 4 main categories. There were microbiomes from pits, from feces. So yeah, kind of gross to think about initially at high school, but it was also exciting. Word was about school that week. Oh, look what we're doing. And dogs, cats, a bearded dragon, we even had a snake. Unfortunately, we were not successful with a snake despite multiple attempts. And actually, I think the feces was a little bit dried out. But it was exciting, and that's for next year. So also water, students who were interested in that collected samples from Lake Washington, Lake Union, Puget Sound. Just a quick anecdote to share with you. Students were so excited that when initial DNA isolation failed, a student went out at midnight one night to go and get more water from Green Lake. So that's kind of what I think makes a project a good level of interest. Soil, and then lastly food. So cheese, yogurt, we had some cheese making and yogurt making families. So they were eager to test their own stuff. Kimchi, sauerkraut, kombucha. So a large part of this was making DNA extraction cheaper and easier. And so normally, if you're in a lab, you would just go to the QIAGEN kit, but we didn't want to do that because of cost. So we tried different methods. We tried the Claremont BioPurelyze, which was a mechanical disruption. We used to kind of pull the sample through some beads with a syringe. This took some practice at first because it's all, if you're in lab and you're used to doing these things, it's a little bit hard, but students could absolutely do this. There was Chelix, which is 100, which is good for simple samples like food. Glass milk worked pretty well. The methods that we ended up, we had criteria that they had to be simple, they had to be economical, and not require centrifugation or expensive equipment. So we do have those little microfuges, which are in many high school labs or classrooms, but I don't have anything that can centrifuge more than that. So DNA extraction is something that is most deserving of trying to optimize some more. We did have to do this step a few times, and with some samples we just weren't successful, but in the end we did extract a whole lot of DNA from different things. There you can see students, and we actually just initially we were thinking one group would do one sample, but we ended up with everybody processing at least 2 because our reagents were kind of cheap, so we could do it a few times and really try to get a diverse set of samples. Okay, and here you can see another slide of students just performing electrophoresis to check their libraries. Which we actually amplified 16S rDNA from our samples, and I guess I forgot to mention, it was also a whole, doing something like this in a whole project in a school, you kind of keep thinking, okay, but they don't know about this, they don't know about that. So metagenomics was another thing that we sort of really wanted students to wrap their heads around, how with normal culturing, you would only be able to see about 1% of what's in your sample, but by actually going for direct DNA isolation and sequencing, we get to see so much more. I believe this is over to you, Chris.
Okay, sure. So yeah, it was quite a eureka moment when the students actually injected the DNA libraries onto the Nanopore sequencer, and data started appearing. As Danielle mentioned, we didn't sequence DNA randomly. We amplified a gene, the 16S ribosomal RNA gene. This is about the most conserved thing in the whole universe. All life on Earth has this, so it's really good for classifying as many different organisms, classifying and counting. And the size of that gene is about 1,800 bases, and there's a little histogram that popped up on the screen as the students, within 5 minutes of the students putting the DNA on the machine, and they could see that they were getting a whole bunch of 1,800 base DNA fragments, or the gene that they made. So they saw the gel before, they saw that peak, they saw it again here. They could see how many reads they were getting per minute on this plot over here. This is a plot of all the flow cells, and the green ones are the flow cells that have DNA passing through them at that instant. So the kids could see that the machine was working. They could see here the fragment size for the DNA, that it was as expected. They could see that some libraries had a lot of full-length DNA this size. They could see some libraries had more fragmented DNA. You want the full-length DNA so that you can most accurately identify the microbial species, but even this fragmented DNA, and some of the samples were pretty fragmented, even that is interpretable. The beauty of sequencing is that you can get information from most anything. So in real time, we used Oxford Nanopore's 16S sequencing tool. They have a web page with a cloud server that collects the data. You can stream your DNA reads to their machine, and you can run it through a quick data processing pipeline. This FastQ 16S pipeline takes it does a quick guess as to the DNA sequence, and then a pretty quick guess as to what organism that DNA came from. It's not incredibly accurate, but it started streaming in 15 minutes after the experiment began. So the same day that the students put the DNA onto the Nanopore, they could start seeing what they had. They looked at the names of the bacteria that were coming out and immediately started going to their books or to Wikipedia to see what those organisms were, and if that was something that they expected to have in their sample. So it really was an exciting moment. But then we wanted to use some better quality tools, and the best quality tools are actually open source, published by academic groups. And so what we did was we used that higher quality GPU enabled base calling. I told you about the machine we made earlier, so that we could more accurately get the DNA sequences, higher accuracy. We wanted to have each base 96 or 97% accurate instead of 92% accurate as it was on the first day. So the next day we had higher quality data, and then we ran it through some other tools that allow you to use different databases to most accurately identify the microbe that the DNA came from. And let me tell you a little bit about these plots. These pie plots are made by a visualization tool called Krona. So each ring of this pie plot represents a level of classification detail. So you might remember from your biology class that Carl Linnaeus came up with these different levels of identification: the kingdom, the phylum, the class, the order, the family, the genus, the species. So here we have the kingdom in the center. That's bacteria. And then we've got phylum, order, genus. Sometimes we have species, sometimes not, depending on the quality of the DNA and the database that we selected. So just for example, let me pick out one of my favorite species. This would be the official state microbe of Wisconsin, Lactococcus. I'm from Wisconsin originally. So here's Lactococcus. It is 11% of the reads, 11% of the pie here on the right. So in the center, we've got the kingdom is bacteria. Then we've got the phylum is Firmicute. And then we've got the order is, let's see, actually, the phylum, let's see, kingdom, phylum, order, class. We've got the order is Lactobacillus. The family is Streptococciae, and then the genus is Lactococcus. This is probably Lactococcus lactis, but this database can't tell us with 100% certainty what it is. So if it isn't certain of the genus, it will, you'll see a stripe here where it'll go as far as it can, but it won't go farther than it can. So I hope that makes some sense. We've got the genus ring on the outside, and the more generic, more general levels of taxonometric order in the middle. So in this case, I've just put up two random example plots. These are two typical microbiomes. I believe these were two dogs. There were quite a few dog fecal samples contributed, and I think this is fairly representative. The most common phylum in all of the dog samples were Firmicutes. Some researchers believe that a high ratio of Firmicutes to other phyla like Bacteroidetes is unhealthy, but it's a little bit more complicated than that. For example, here you'll see within the Firmicutes, we've got 11% of the total reads are Lactococcus, which is usually considered a good bacteria. It's a probiotic, often introduced into foods, into food cultures. It competes with Staphylococcus, Clostridium, Salmonella, and a few others, all for the same sort of resources. So often if you have one of those bacteria increasing, you'll have the others decreasing. Now, some species of Staphylococcus, Clostridium, Salmonella, etc., are pathogenic. Students were very interested when they heard the name of a potential pathogen, but they also noted that only certain subspecies of each of these actually cause disease. And so you really need more sequencing beyond just the 16S gene to know whether the particular bug you have is pathogenic or not. But they were very interested to see that some of the other pets had higher counts of potentially pathogenic species like Salmonella. The bearded dragon sample was particularly high in Salmonella, 4%, but it was that very healthy lizard. Students found that cat and dog microbiomes were quite distinct from each other. One group looked at outdoor cats versus indoor cats, or outdoor dogs versus indoor dogs, and the outdoor pets did not resemble other outdoor pets, and the indoor pets didn't resemble other indoor pets. Rather, it was the host species. The dogs were quite similar, and the cats were quite similar to each other. These microbiome samples taken from pets were quite diverse. This one on the left, although it's almost entirely Firmicutes, it's quite a few different Firmicutes. This one on the right is very diverse. Other samples like water were quite diverse too, with a lot of overlap between the water samples and the pet fecal microbiome samples. Comparing the lake one group compared lakes to Puget Sound, or lakes to freshwater lakes to Lake Union, which is rather brackish. All of them were quite complex. Green Lake was found to have a much higher level of blue-green algae than Lake Union. Students theorize as to why that may be, and also noted that visibly there is a lot more algae in Green Lake, hence its name Green Lake. Some students went beyond just the bacterial databases. Some students also looked at eukaryotic databases. So that would include yeast and multicellular organisms. They found that PET samples might often contain fly DNA. So with that, they could speculate that from the time the sample was deposited in the litter box to the time it was collected, it was probably visited by a fly laying eggs. With food samples, like the pickled samples, they could find vegetable DNA, for example, cucumber chloroplast reads. So that's just an example of some of the observations that the students made. Many groups did many pairwise comparisons, so really there's too much to talk about here, but I just wanted to show you how they got started. Okay. Here's an example of a few of the posters. I'll turn things back over to Danielle.
Thank you. So yes, this is just to give you an idea of what the posters looked like. They were very creative with their names. There's the lab report for the dogs. And this was actually one of our practice sessions. If you can go to the next slide, Chris, please. Here is on last week, Tuesday night, we had our first public exhibition of learning at Lincoln High. And so we had all the parents come through and community members and students prepared how to share their learning, which again is a skill to be able to talk about science. And they had a lot of excitement, and they even made a large greetings from the microbiome selfie wall, which we are featured there as a picture against the selfie wall. So lots of feedback from the public, lots and lots of excitement. If you can go to the next slide, Chris, thank you. Just to summarize our findings, so two major take-homes. First, that DNA sequencing is no longer too expensive to consider. So glass, milk, and fungal methods dramatically reduce the cost. And that DNA sequencing is perfect for project-based learning, which is a better way to teach science because it is using within all the content, students get to propose and ask their own open-ended questions. So they're doing what they call real science. And that I'm also really excited about continuing this project next year with a new batch of students, where I'm thinking of starting off with looking at our posters and kind of thinking about what are questions they asked, what are questions worth pursuing, and where we're going to go from there. So overall, very successful.
All right, I'd like to thank some of the people that helped us out with this. So first, of course, we needed Toshiba. We're very thankful for their contribution to purchase equipment and reagents. I want to thank all of the genome science students who participated. It was great working with them. Jeannie Chowning and Regina Wood at the Fred Hutchinson Cancer Research Center Science Education Partnership provided some loaner materials and reagents for testing different extraction protocols. We also got some materials from Mag Biogenomics and some good advice from Eric Tong at high school in Hawaii. On the right is a number of ISB scientists who provided consulting or materials for testing different methods. So a lot of people all contributed, and I think we can get more help from them in the future as well. Especially as we work to try to expand this to more and more schools. All right, well, thank you so much for your time. That's all we've got for now, and so we'd be happy to take some questions.
Thanks, Chris and Danielle. That was great. I love the lab report. So folks, just a reminder, if you want to pose some questions, please use the Q&A box at the bottom of your screen and just type your questions in. And I'll get us started off here. Yeah, Danielle, I'd love to hear from you a little bit more. You know, you mentioned right up front, the kids want to be real scientists. They want to do in the classroom what people like Chris are doing. Can you talk about the importance of that and maybe why that's so novel?
Right. So that is, like I said, this was something that students pointed out to me every day, that they feel they're doing real science. And so even though we do really exciting labs and phenomena-based teaching, so we typically don't just say, okay, now we're doing PCR. We go, okay, we're going to look at, genetically modified food or so on. There's still a sort of an expected outcome. So, usually with a lab, we say to students, oh, look, we're doing science. You're scientists, you're doing science, but they can tell the difference between actually doing a project where from the beginning to the end, they are not sure how long it's even going to take because they don't know what problems are going to come up, and they are going to have to troubleshoot, and they are going to have to maybe change course. And so the more we sort of struggled in a way, the better it went. And students just really were also very excited to do things that are in the community. So things to do with them. And when they have a voice in asking the questions, then it becomes really important. Another aspect which we really try to do is incorporate social justice into some of the questions they ask in science.
Yeah.
And I just want to give a shout out also to Jeannie and the folks from Science Education Partnership, which are also involved in that work. And students really, to be engaged, they want that connection to not just in the classroom to learn the content. The goal needs to be to do real science or find things out and learn a process.
Yeah. Okay, here's a question. Do you have reading materials you recommend for students of this class to get them familiar with the terms and concepts?
Yeah, so that's actually a hard one. So we do officially have a textbook for this class. It is Introduction to Genomics, but I found that this textbook, the level was a little too hard actually. And I don't want to make this course something that is just accessible to students who have already taken AP Biology. So pretty much I am scrounging around for readings, and I have found actually things like, for example, in popular science books by people like Carl Zimmer and Ed Young and so on, that's a great way to talk about things like the microbiome. And then in terms of other readings, I just kind of hunt around. I often we look at papers and we just look at figures from papers and we try to sort of talk through them. And that really helps a lot. So it is it's a rigorous science course, but it is an alternative to AP, because I feel that's really important to be equitable and provide access to all students to learn about these things without having to feel AP has a high bar in terms of homework and the test and all that and pacing.
That's great. Thank you. So another question is, this seems like a unique situation. You used to be a scientist, you're married to a scientist, and the two of you are super passionate about this, and you've created this fabulous program. Is there a way to replicate it and share this model with other teachers and schools who might be interested?
Well, I believe there is, and the danger, of course, is you don't want to make it too prescriptive to sort of become a kit that just you follow a recipe again, right? So to have the technology available. And I think we're not quite there, but I think we are moving that way. We need to optimize the DNA extraction methods to make that work a little bit more reliably. But I think it would be amazing to work towards having a kit that can be checked out. Some other challenges are that these flow cells have a shelf life. And so the more people are able to do this, in some ways it will be better because we can order things together and distribute it. And so, so I do think it would be fine. There is a model out there where I know scientists are amazing at working with teachers at high schools. And this would be another incredible thing, because I think the day of loading the flow cell is kind of nerve-wracking. And, you know, that's a great way to donate an hour to come to a classroom and just kind of give that teacher and the students peace of mind, because it's kind of a big deal, you know, to load all of your library on the flow cell and see it go.
Yeah, it seemed like a big deal. And I love what they were studying too, just indoor cats versus outdoor cats. I mean, that's interesting. I never thought about that. So that's really great. We do have someone who's just volunteered to help you put together materials. So this is paid off for you. You're already recruiting a team.
Fantastic.
Yeah. And actually there's someone else asking about volunteering. Are there volunteer opportunities to help out during the semester?
Well, I'm at one school, and I also am not quite in a position to take on to distribute this widely. But I believe ISB would look into that, and I believe that the Science Education Partnership at the Hutch would also look into that, and for this and other high throughput techniques. So so there's always ways to volunteer. I mean, I think that would be an amazing thing to do. In terms of volunteering in terms also of making this accessible in more schools, we need a place for it to land in courses, and that is difficult. So most schools have, or lots of high schools have, biotechnology courses, but we also need excellent teacher professional development. And that is something that ISB is amazing at, providing opportunities for teachers to come and work with scientists in the summer. And I would think that this is something that would be a wonderful way in which ISB could have teachers come and get familiar with sequencing.
Mm-hmm. Yeah, we love fostering that next generation of scientists, and we also just want our youth to be scientifically literate, right? And especially after a few years of a pandemic, it's really important that people understand and appreciate science, and because I think if you understand it, you can trust it too, right? That's a huge issue lately. So.
Absolutely. And I think it's incredible that ISB brings all these young people in to the Institute and works with them. And I know you've recently expanded how many students you can bring through and have these opportunities. One thing is, though, that what students need to get to ISB, get to the point where they apply for that. And so my dream is to catch people in school and provide more of these opportunities in a school setting with access and opportunity to kind of learn about ISB, or maybe go there for a field trip or whatever, so that we can just get more students excited and create access to this world of biology for more students.
Yeah. Well, and as I was listening to what you're putting your students through, it seems like the only thing you're not teaching them is how to apply for an NIH grant. It seems like I think you have everything else covered about being a scientist.
We do talk about how expensive it is to science.
Okay.
And who gets grant and who gets to pay, and you know, who gets to ask the questions, and all those things are very much talked about as well. Kids want to know. Kids know a lot.
Yeah, they do. They do. Yes. What you're doing doesn't look anything like what I was doing in my high school science classes.
Me neither,
so I'm envious. Yeah. I mean, it's really great. Well, thank you very much. It was great of you to spend this time with us. And we had a really good group today. There were a lot of people interested in this topic. So we're happy that you joined us. And thanks to everyone else. Thank you to our guests today for joining us. If you're on our email list, you'll get more information about events just like this. We try to do them almost once a month. And so keep an eye on your email inbox. You can also go to the events tab on our webpage, and you can look at past. We keep all the recordings of these research roundtables on that webpage, so you can surf a lot of different topics and look over those. If you're inspired to make a gift today to support our STEM education work, we would gratefully receive that. You can go to isbscience.org and click the donate button. And that's all we have for you tonight. Thank you so much for joining us. We really appreciate it. And we'll see you again in the future.
All right. Thank you.
Thanks,
Bye-bye.
Rose. Thanks, Danielle.
Thank you so much.